Howdy folks, thanks for checking in to MrUlrich's I'm Mr. Ulrich. We've been talking about enzymes. In this little notes cast, we're going to talk about how to keep those potent little proteins doing what we need them to do at just the right rate. So we've been discussing how enzymes are important molecules. They make life possible by lowering the activation energy needed for metabolic reactions to actually take place. And the beauty of their catalytic reactions, catalytic abilities, comes from the fact that they don't get used up in the reaction. If you would imagine, if you sit down and eat a hamburger and then you need to synthesize a new protease for each molecule of protein that you digest, it would certainly be a losing situation. So enzymes, being biological catalysts, are effective at really, really small amounts. It doesn't take a lot to get a big effect. And being so powerful, they need to be controlled. They need to be allowed to work sometimes and not allowed to work at other times so that metabolism can happen at the right rate. So how do we do this? Probably the most basic level of enzyme control is genetic or epigenetic, and that would involve turning on and turning off genes so that we can control when the enzyme is being synthesized in the first place. This can happen at the nucleosome level, where in order for a particular gene to be transcribed, it has to kind of unwind off of its histone molecules a little bit so that the RNA polymerase and other transcription factors can get in there. And so in order for this to happen, those nucleosomes have to have a little bit of distance and be a little loosely packed. Through alteration of the histone tails, we can cause, and other parts of the histone molecule and DNA molecules, we can cause those nucleosomes to kind of clump together, not give enough room for the transcription factors to get in there and turn off the gene. Another way of altering transcription is through methylation of cytosine, and this is one of the more permanent ways of keeping a gene from being transcribed. It still maintains, all of these methods still conserve the genetic code, so it doesn't change the order of the bases. It just turns on or turns off the gene. Enzymes can be controlled a little bit more directly through the presence or absence of other molecules that are going to interact with the enzyme, binding with it, either right at the binding, the active site itself, or some other portion of the enzyme, but are going to modify its ability to interact with its substrate. Some of the things that can affect enzyme action are inorganic, like iron. Iron makes up the heme group of hemoglobin. There are 4 heme groups in each hemoglobin molecule, and their ability to transport oxygen certainly affects electron transport, which oxygen is part of, through binding with oxygen and being able to drop off oxygen as well. So it makes a really good oxygen carrier. On the organic side of the cofactor tree are the coenzymes. These are non-protein still, but they're organic. These are compounds that temporarily bind to an enzyme to give it its proper shape. In this example here, the cofactor actually makes up part of the active site by bonding to the protein part. We call that an apoenzyme, the protein part, and then you add the cofactor and you put it together, you get the whole enzyme, the haloenzyme. And that cofactor gives the enzyme the properly shaped active site. A lot of vitamins act as coenzymes. There are prosthetic groups as well. These are kind of like coenzymes, except these bind basically permanently to the apoenzyme in order for it to work. The heme group is a good example of this. The heme is permanently part of the hemoglobin. Cofactors are promoters of enzyme action. That is, their presence actually helps the enzyme do its job, or makes it even possible for the enzyme to do its job. There are other compounds that, just by their very presence, they inhibit the action of the enzymes, basically by getting in the way. One form of inhibition is called endpoint inhibition. This is also known as negative feedback. Metabolic pathways typically are made up of several different chemical reactions kind of in a chain, where one compound A turns into B, and then B is converted into C, and then C is converted into D, and E converted into F, and blah, 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 blah. And each one of these chemical reactions is controlled by a specific enzyme. In endpoint inhibition, the product of the chemical reaction, in this case compound F, actually feeds back on one of the enzymes here, it's enzyme A, and will inhibit its action. And that way, the more F there is, the less B there is produced, and that will slow down that chemical reaction. Once there isn't as much F around, then the enzyme A is disinhibited, it's turned back on, and then it makes B and C and D and E and F. Another form of inhibition is called competitive inhibition. Here, if the active site here is shaped just right for this particular substrate to go through the chemical reaction and break down into two parts. Inhibitors have the same shape, the same, and are allowed to bind the same way to that enzyme, and thereby block it up so that the substrate can't get in there. A lot of neurotransmitters work on this level. Another form of inhibition is called allosteric inhibition. Allo for other and steric for site. These are compounds that actually act as kind of like switches. See, we've talked about the active site where the substrate fits. Enzymes can also have another place where other compounds can fit, which we call the allosteric site. Now, these aren't directly involved in the chemical reaction itself, but what they do by bonding to that allosteric site, they cause a conformational change that's reversible. So they cause that conformational change that changes the shape of the active site so that it can no longer bind. And then when there's less of that material around, that allosteric inhibitor around, then the active site changes back to its regular active state and can bind to the substrate, and the chemical reaction is turned on. This is called non-competitive inhibition because it's not competing for the substrate for kind of access to the active site itself. Phosphofructokinase is a great example of an enzyme that is controlled both through allosteric inhibition and endpoint inhibition through negative feedback. Phosphofructokinase is part of cellular respiration, that cascade of chemical reactions that produces ATP. It's important because it changes one compound, fructose 6-phosphate, through phosphorylation into fructose 1,6-bisphosphate, which is kind of the next compound in the sequence of reactions. So as respiration takes place, we get a buildup of ATP. ATP also fits right into an allosteric spot on the phosphofructokinase. And in doing so, it actually changes the shape of the active site so that fructose 6-phosphate, one of the substrates, and ATP, which happens to be also the substrate, not only does it fit into the allosteric site, it also can fit into the active site. Once ATP fits into that allosteric site, those substrates can't fit in there. Without the substrates, of course, the cascade of chemical reactions can't proceed and respiration slows down until there's less and less ATP in the cell. These bonds at the allosteric site, remember, are temporary, and so as the concentration out in the solution, in the cytosol of ATP goes down, the ATP will break off of the allosteric site, opening up the active site again, and the respiration proceeds. course, it is possible to put those things together and get competitive allosteric inhibition. In this case, um, the inhibitor fits into an allosteric site, but by fitting into that allosteric site, it actually blocks off the active site. Kind of nasty, right? Getting in the way of everything. Loves me some fruits, but hate it when they get all brown and gooky. Now, my grandmother used to put lemon juice on her sliced apples before she put them in the pies to keep them from getting all brown before she got them in that pie there. So here's a little experiment, a demonstration showing how different compounds, like lemon juice for that matter, can control the enzymatic action that's involved in browning fruits. So in this little demo, there's a control where there's nothing there, it's just open to the air. And then we put some of the apples in water. They put some of the apples in acetic acid, which is basically vinegar, and citric acid, which is the lemon juice that this was done using regular citric acid, but lemon juice provides the citric acid, and ascorbic acid or vitamin C. So here's the control. See, it gets nice and brown after 20 minutes. Leave it a little bit more time, and it'd be pretty good. When we look at the water, however, compared to the control, the water seems to, I mean, it doesn't look too brown there in the end. It seems to have done pretty well. Likewise with the vinegar and the citric acid, right? No browning. And the ascorbic acid. So all of these compounds, water included, seems to be able to keep the enzymes from breaking down the molecule. The important thing for us is not just that lemon juice is effective in lowering the browning of fruits, but the hows or the whys these enzymes are kept from their jobs. Well, in the case of vitamin C, it's an antioxidant, and it provides the oxygen something even more attractive, say, for it to work on rather than for it to oxidize rather than the delicious phenolic compounds of apple. The citric acid is actually a chelating agent. That means that it kind of surrounds and binds up the copper ions that normally would be there and allowing the enzymes to do their job. No copper ions, no enzyme activity. Acetic acid actually changes the shape of the enzymes themselves by reducing the pH. Once you add the acetic acid, those enzymes are done. And even the water, just by itself, was effective. And it does its action basically by just blocking the amount of oxygen that can get to the apple in the first place in order for that to take place, in order for the oxidation to take place. We'll stop there for now. Thanks for checking in. Again, I'm Mr. Ulrich. Any questions or comments can be emailed to me. You can also go to Mr. Ulrich's Land of Biology if you didn't get here from there and see what's coming up in class and what we've been doing and all kinds of other tasty little tidbits over there. So again, thanks for checking in, and we'll see you in class.